Whenever you guys perform molecular docking, you start from protein ligand complex. The ligand present in your protein molecule gives you idea about the active site of your protein. But there are instances when you have to start your molecular docking from a apoprotein, that means the protein without your ligand molecule. And in that case, how you can find where is the active site of your protein? And in order to find that, you should follow this tutorial. In order to find the ligand binding site in your protein, we will use the Autoligand program, which is already included in the AutoDockTools. We will start this tutorial by downloading our protein. We will again go to the PDB database. We will now open the PDB database to download our protein structure. 3B7E is our protein, and we will download its PDB format. We will visualize this our protein file in Pymolling. As you can see, it has a bound ligand, which is actually a zanamivir, and this drug molecule is in complex with our protein. Now, the drug that is binding in the protein is actually present in the active site of the protein. So, with the help of Autoligand program, we will find the we will predict the ligand binding site of this macromolecule and check whether our ligand binding site is present in that uh, predicted ligand binding sites or not. And in this way we will confirm that the Autoligand program is giving correct prediction of the binding sites or not. We will now open the AutoDockTools and prepare our protein. We will set the path to our working directory by going to preferences set, and we will replace the start directory path, startup directory path to our working directory. We'll copy this path and we will paste it here. We'll click on set. We will dismiss it. Next, we will load our macromolecule in the molecular viewer window of the AutoDockTools by going to File, Read Molecule. We'll open this PDB file that we have just downloaded. This macromolecule has three chains, that is A, B and C. We will keep just the B chain because that's where our ligand is binding, and we will remove the chain A and chain C. We will select chain A and we will select chain C, and we will remove them by going to edit, delete selected items and click on continue. We have successfully removed the unwanted chains and kept the P chain. Non-protein molecule, which is basically the drug molecule which is already present in our protein structure. In order to select that drug molecule, to remove it, we will go to select and click on select from string. In the residue section, we will type ZMR, which is a short name alliance for our ligand name, and we will click on add. You can see here in the molecular viewer window that our drug molecule has been selected by the yellow crosses. We will dismiss the dialog box. And next we will again go to edit. We will click on delete and we will click on delete selected atoms. Click on continue, and you can see our ligand molecule has been removed from our protein structure. Next, we will remove the unwanted water molecules that are associated with our protein structure. These water molecules are actually crystal structure associated water molecules, and we need to remove them. Therefore, we will go to edit. We will click on delete water, and you can see that the red dots which represent the oxygen atom of the water molecule has been removed. Next, we will add hydrogens to our protein structure. We will go to hydrogens and we will click on add. In this case, we will add all hydrogen atoms rather than adding polar hydrogens only. In the previous docking tutorial, we have added polar hydrogens to our protein structure instead of all hydrogens. But whenever you want to find ligand binding sites on your protein molecule, you should provide all hydrogen atoms along with the polar hydrogen atoms to your protein structure. We will accept all the default values and click on OK. Here you can see in the molecule viewer window that hydrogen atoms have been added to our protein structure. Next, we will go to grid and we will write the PDBQT file. Grid, we click on macromolecule and click on choose. A dialog box will appear and we will choose our 3B7E protein from the dialog box and click on select molecule. Click on OK. Provide the suitable name to your protein. Here I will specify the name as protein dot pdbqt. Make sure that you provide explicit file extension after the file name. We will go to our working directory and see whether our protein pdbqt file has been prepared or not. So you can see here the Autodock structure file has been prepared, and we can confirm the pdbqt extension by going to properties section. So here you can see .pdbqt extension is present with our protein file. Click on OK, and here we have completed our first step, which is preparation of our protein structure. Now in order to run Autoligand, you have to carry out two pre-steps, which is the generation of the grid parameter file and affinity maps. So Autoligand requires two input files. One is the grid file, which you provide as a search space for the Autoligand program to search over the provided surface for potential ligand binding sites. In grid parameter file, we will actually provide a complete surface of the protein, which Autoligand program can utilize and search for potential high affinity ligand binding sites. But it also along with the grid parameter file, it also requires affinity map file, which consists affinity of amino acids of your protein structure to the atoms present on your on your potential ligand molecule. Actually, ligand molecule consists mainly of the carbon atom, then the hydrogen acceptors and the hydrogen donors. So we need to check which amino acids on our protein structure or amino acids on our protein structure have high affinity towards these three groups of atoms, and that binding energy, binding affinity values are written in a separate file, which we call it as affinity map files. So one by one we will generate these two input files so that we can provide them to Autodock and generate we can provide it to Autoligand and generate the potential ligand binding sites. So first we will generate grid parameter file, the Autoligand, or we need to provide the AutoDockTools with which map types we will work on. Therefore we will go to grid, then we will open set map types, and then we will click on directly. Here you can see one dialog box has appeared, and here you can see there are 7 atoms against which Autoligand can potentially find binding affinities of amino acids. But here you can see carbon is represented as C, hydrogen donors are represented by HD, and hydrogen acceptors are represented by OA. So we will keep only these three groups, and rest we will remove. So we will remove it one by one, and we will click on accept. Now we will generate the grid box, and we will enclose the complete protein structure within the grid box. So we will go to grid, we'll click on grid box. It is really important to set the spacing of this grid box to 1 angstrom. Here it is actually By default 0.375 value is set. 0.375 value is set for the spacing. We have to change this value to 1 angstrom. For that, we will right click on the gray box and one dialog box will appear. The value of this gray box spacing is set to 0.408. We will change it to 1. Then click on apply. Now you can see in the molecular viewer window that the grid box has enlarged in size. We will dismiss this. Now we will change the number of points on the X, Y and Z dimension. For that we will again right click on the red thumb wheel and the initial value is set to 40 by default. We will change this value to 60. We will actually keep all the values of X, Y and Z coordinate as 60, 60, 60 only, as it will be sufficient to cover the entire protein. Again we will right click on green thumb wheel button on the top and we will set its value as 60. Now we will finally change the value from 40 to 60 of the blue thumb wheel. Dismiss. In the molecule viewer window you can see that the grid box has completely covered our protein structure. Now we will go ahead and go to the file section of the grids option dialog box. So we'll go to file, we will click on close saving current. Now again we go to grid and we will go to output and we will click on save GPF. Provide the meaningful name to our grid parameter file which is of .gpf extension. The GPF stands for grid parameter file. So I will give here the name as grid.gpf and click on save. We can go to the working directory and see that our GPF file has been created. So these are the contents of the grid parameter file. It has the X, Y and Z coordinate of our grid box. Then it has the grid data file which is of the name protein.maps.fld. Then we have the information on the spacing and the map or the affinity maps against which atoms we are searching that should be or might be present in the ligand are carbon, then hydrogen donors, then hydrogen acceptors, and the name of the output file they have been specified here. So for each atom one affinity map has been generated will be generated once we run our Autoligand program. For example, we have chosen a carbon atom against which we will check that our amino acids have affinity or not and at different places over the protein structure. Therefore the output of this so the output of these information, that is binding affinities, will be stored in the map C map file, c.map file. Then similarly the affinity against hydrogen donors will be stored in HD map files and OA will be stored in OA.map file. Apart from these three affinity maps against the specified atoms, there are two additional map files which are electrostatic potential map files and dissolution map files, and these 5 files will be the input to our Autoligand program along with the GPF files. Now we will go to the command line, but before going to the command line we need to bring the AutoGrid program to this current working directory. AutoGrid file can be found in the Scripps Research Institute folder which is created in the program files folder that is present on your C drive, and you can go there and you can copy the file which is the exe file and you can paste it in your current working directory. so you don't have to provide an explicit path while running the AutoGrid command. We will open this folder on the command line by typing cmd in the path ribbon. So as you can see, we have opened this particular folder, which is our current working directory in the cmd. So we don't have to provide an explicit path to any of our files. So we can just write the name of the program and directly write the name of the input files without providing any explicit path. So the current program we are running is the AutoGrid 4 program. Actually, this program converts the GPF file to GLG file, which is grid log file. So we will write AutoGrid 4 .exe. We should provide the .exe uh, extension of our following the AutoGrid 4 name, because it is actually a executable file, and we should tell the command line about the file type of the program we are running. So -p is, here we are giving the parameter file as the input to the grid 4.exe program, so -p, and we will write the name of our parameter file, that is the grid parameter file which we have just saved. So the name of my grid parameter file is grid.gpf, sorry, and we will provide the name of the log file that will be our output file. So -l stands for log file as the output, and we need to provide the name of the log file that is being outputted, that will output after we run the AutoGrid 4 program. So grid.glg, and we will run this process in the background. So we will add the ampersand operator and hit enter. It will take some time to run. So as you can see, the glg file has been created in our working directory, and we will open it. And as we go to the complete end, we can see that the execution has of the AutoGrid 4.exe program has successfully completed, and you can see different map files has been generated. Now we have converted our GPF file to GLG file and set our map types to carbon, hydrogen donors and hydrogen acceptors. We will now move ahead and run Autoligand program. For that, you need to first download the autoligand.py file. This autoligand.py file is actually a Python file which you can obtain by going to the GitHub page of the AutoDockTools. I will show you how you can download this. So we will again go to our browser and we will type here autoligand.py file. Go to the first link, which is actually a GitHub page. Here you can see this is the Autoligand file written by the Rodney Harris, and we will go and download this raw file in our current working directory. So yes, we will go and save. We will return back to our working directory, and again we will confirm that the Autoligand program has downloaded, and you can see it here, Autoligand Python script, and we'll again go and check its property for .py extension. So click on OK, as the .py extension is actually present. Now that we have successfully created our map files as well as the grid file, we will now go and run our Autoligand program. But before that, make sure that autoligand.py file is located in your folder along with your map files and the grid files. So we will now launch our cmd by typing cmd in our location ribbon. We will again list the files present in our directory with the dir command. So as we can see, map files are present, the ligand file is present, and the glg file is also present. So now we will run the command python auto ligand dot py. We provide all the map files with the dash r tag, and we will write the just the front portion or the starting part of our map file name. So as you can see, there are three map files which we have generated for the carbon affinity map, then we have generated for the hydrogen affinity map, and we have generated for the oxygen affinity map, or the hydrogen acceptor affinity map. So as you can see, all these map files have their name starting with protein. So we will just provide this protein as the starting name for our map files. So we will write protein, and we will provide the search space with the help of dash p tag. So we are providing the search space of just 100 atom long, and we will hit enter. This process will take time for about 10 to 20 minutes to run. So as you can see, the process is running, and it will at last generate uh, 10 binding site, or the 10 binding sites, which will be stored in a single pdbqt file, which we will load in our AutoDockTools along with our proteins crystal structure, that is pdb file. So as you can see, our Autoligand program has successfully finished its run, and it has generated 10 new structures. Actually, these structures are the surfaces where Autoligand has found, which can be our potential ligand binding sites, and it has outputted, as you can see here, it has outputted 9 pdbqt files which we can visualize. Moreover, it has also written the total energies and total surface area of each of the binding site that it has found. As you can see here, the first pdbqt file, which is this file, uh, has has the total volume of 270 and its EPV value, which stands for energy per volume value, it has given to us. As you can see, there is one peculiar thing about these values. EPV values of structures 3 to 9 ranges from the value of minus 0.18 to minus 0.14. But you can see the first two structures, or the first two ligand binding sites, have exceptionally low EPV values. Also, they have very low surface area, or the very low total volume, which is up to 270 and 276, while the other structures have the total volume that ranges from 300 to 334. Although there are some values where the total there are some structures where total value is total volume is 260, which is lesser than the first two structures. This exceptionally low EPV values suggest that these surfaces, or the volumes, are actually present on the surface of the protein, or they might be potential water binding sites. But these can be decided upon visualization of these two structures in our Pymol. So we will now go and open each of these surfaces that are generated by our autoligand algorithm, and see where these surfaces are actually located on our crystal structure of our protein. So we will open our crystal structure of protein, and we will strip out its chain A and visualize only the chain B along with the surface waters and the ligand ZMR in Pymol. So let's open our protein's crystal structure in Pymol. So this is our crystal structure of our protein, and this is chain A. As you can see here, I have selected one amino acid, that is valine 290, from chain A, and on the right hand side you can see there is chain B. So we will strip out the chain A, which is present on the left hand side of our molecular viewer window, and we will just keep the chain B. So how we can select the chain A? It is very simple. We will write a command select. We will give a name to our selection, for example chain underscore A, comma. We will set uh, the name identifier on what basis we are selecting the chain A, on what basis we are selecting the atoms in our protein structure. So we are basically selecting the chain A, so we will write the chain identifier chain, and we are writing the chain name or the chain ID that is associated with each and every chain in the PDB file of the protein. Therefore, the chain identifier here is A, so we will write capital A and we will hit enter. So as you can see, Pymol has selected 3517 atoms from chain A, and it has stored all these selected atoms in our provided name, that is chain underscore A. And in the molecular viewer window you can also see that the selected atoms are highlighted in pink dots. Now on the right hand side you can see there is new selection being made, which is chain A, and it is associated with the parenthesis. So we will go to the hide button and we will click on everything. So as you can see, we are only left with chain B, and here you can see our ligand molecule is associated with our chain B of our protein structure. Now one by one I will open the surfaces that are generated by the Autoligand program. So we will open the first fill surface. We'll go to open and we will open the first fill surface. This is the surface the Autoligand has generated, and as you can see, this surface is present on the surface of the protein, and hence it is more occupied by the water molecules. Although the representation is in stick, we can convert this representation to surface format in order to visualize the actual binding surface the Autoligand has generated as its first pose. So to do this, we will go to the show button of the fill 1, and we will go all the way to surface, and here you can see that the stick representation has been converted to the surface representation. Now we will hide the underlying stick representation. For that we will go to hide and we will click on sticks. Now we want to see uh, how many water molecules are present inside this surface, because this surface is actually present on the surface of the protein and it should be occupied by the water molecules. And to do this we will adjust the transparency of this surface representation by typing the following command: set transparency, comma, we will set that transparency uh, from initial transparency of 0 to 50% transparency. So we will write 0.5, which represents 50% transparent as compared to the initial transparency level. So the initial transparency is by default set to 0 for the surface representation, and we will convert this uh, 0 transparency to 50% transparent surface, comma, we will specify which group or which selection we have to make transparent. So we have to make the fill first representation as transparent. So we will type out the name of the selection, that is fill underscore protein underscore 100 out comma 100 out 01. So here you can also start with the typing the name of the file that we have loaded as surface, and you can press the tab button and it will automatically fill up the selection name that matches with the words you have just typed. So here you can see on the right hand side that there are 5 selections that are present. One is all, one is 3B7E, one is chain A, one is SELE, and one is fill underscore protein underscore 100 out 01. So if I want to select the fill selection, I will start by typing out fill underscore and I will press the tab button, and it will automatically complete my selection name. Now we will hit enter, and as you can see that the surface representation have now become more transparent. Now as you can see, the stick representation is not yet hidden, visible, and we were hiding the stick representation. So we will go to hide and we will click on wire. So it will hide the wire representation and we will only left with the surface representation. Now as you can see, the water molecules are represented as small dots, and we want to represent these water molecules as uh, small spheres. So we will change the representation of the water molecules from dot to spheres. For that we will need to select all the water molecules, and we will write the following command to select the water molecules. So select, give the name to your selected atoms as water, comma, we are selecting the water molecules with the help of their residue names. Uh, Water molecules are represented as HOH as a residue name in the PDB format of the protein structure. So we are selecting on the basis of residue name. Therefore, we will write RESN, and the name of the water molecule is HOH in PDB format. We will press enter, and here you can see in the molecule viewer window that the pink dots are the selected water molecules, and in the console you can see Pymol has selected 947 atoms, and it has stored it in the water variable name or the selection name. And on the right hand side you can also see one more selection has been added, which is of water, which is named as water. So we will go to the show button and we will go all the way to spheres. So as you can see, the water molecules have been represented as red spheres, but as you can see the size of the sphere is too large. So we will reduce the size of the sphere with the help of the following command. So we will type set sphere underscore scale, comma, we will scale down the sphere to from its original size to 25%, so 0.25. Also provide the name of the selection, so water, and we will hit enter. So as you can see the sphere size has been reduced from its original size, which if we consider is 100%, and now the sphere size has reduced to 25% of its original size. Let me make the surface representation less transparent so we can clearly see the generated surface. For that, we will again write the command set transparency, comma, 0.35. We are making the surface less transparent, so we are placing the value 0.35, which is more nearer to 0. That will make our surface representation less transparent, comma, we need to specify the name of the selection, that is fill, and with the help of tab I am completing the name of the desired selection. We'll hit enter, and as you can see the surface has become less transparent. We can also make it less transparent by setting it to 20%, 0.20. Click on enter. So as you can see here, the surface representation has consist of significantly greater number of water molecules. So the surface encloses approximately 8 water molecules. So 1, this is the first water molecule, this is second, third, fourth, fifth, sixth, seventh, yeah. So it has enclosed approximately 7 water molecules, and this surface is actually uh, present near the surface of the protein. So this is actually an internal water fill site, or the water space inside the protein, which is nearer to the protein surface or nearer to the protein surface. So this surface cannot be considered as the ligand binding site, as it is mostly occupied by the surface waters associated with our crystal structure. Now we will open our second uh, predicted binding site that the autoligand has generated, and see whether it overlaps with our ligand or not. We will search for such a predicted binding site that overlaps with our ligand, so that we can confirm that autoligand does indeed predict the binding site. So as you can see, I have loaded all the remaining surfaces and adjusted them in surface representation and made them transparent, as we have done for our first fill volume. So we will look at each surface one by one, starting from the second one. So I will hide rest of the fill volumes, so only second is visible. So as you can see, the second volume I have represented it in blue color. So here also you can see the volume has enclosed many water molecules inside it, and we can count those water molecules as well. So this surface has also enclosed approximately 8 water molecules, and it is also present near the surface of the protein. Actually, this surface is overlapping with our first fill volume surface. As you can see, the first fill volume surface I have represented it in a red color surface, and the second fill volume I have represented it as blue color surface. So as you can see, both the surfaces are overlapping, and they enclose approximately 7 to 8 water molecules, and they are also present nearly on the surface of the protein. So these are internal water filling sites of the protein and cannot be the ligand binding site. So we will hide both these surfaces, and we will now reveal the third surface. The third surface again is a water fill site, which I have represented it in maroon color. Here also you can see the surface has enclosed many water molecules. Although this surface is present inside, that means the core of the protein, but still it occupies or encloses many water molecules. So this is again a internal water binding site. Now we will move on to the fourth surface. Here you can see the fourth surface. I have represented it in cyan color. It completely encloses our ligand molecule. So as you can see, this is our ligand and this is the fill surface or the fill volume, and it almost has enclosed just the ligand portion, and it is our ligand binding site. And here you can see there are very less water molecules enclosed by this surface. Just the two or the one water molecule is only enclosed, and it is present inside the protein surface. Moreover, it has enclosed the area where the ligand is actually present. So this is the potential ligand binding site, and its EPV value, you can see it lies in the uh, exact range, and these values are not, and this value is not an outlier like the first two value, which is 0.20, and these two values are actually the outlier, as you can make out from the series of the 10 values or 10 to 9 values. So the fourth EPV value is nearly at the median, and as you can see it encloses just the ligand, and it has a volume, that is fill volume of very less, which is 279, which is approximately 280. So uh, moderate EPV value, or which is lying near the mean EPV value, and an enclosure of very less water molecules, and at the same time very less or suitable total volume will be your binding site, or the corrected binding site, or the ligand binding site the autoligand has predicted. As you can see, the fifth volume is present exactly on the surface of the protein, and it will enclose the maximum water molecules. So this cannot be the ligand binding site. Now we will close the fifth and we will go to the sixth. Sixth is again the internal water site or water occupying site, so it cannot be the ligand binding site. We'll move to the seventh, and as you can see, the seventh representation overlaps with our fourth representation, which is the potential ligand binding site. So we will hide the sixth, we will hide the fourth, and you can see it has also enclosed our ligand molecule. Moreover, it has some extended volume that is occupied by the seventh pose as compared to the fourth pose. So fourth pose has limited its volume just to the ligand, whereas in the seventh pose you can see the volume has been extended to the inside of the protein. This extended volume of the seventh pose is again the internal water structure, and this upper portion of this site can be ignored. So seventh and fourth both are our potential ligand binding site. So I will hide the seventh, I will hide the fourth, and we will go to the eighth pose. So here you can see the eighth surface is again overlapping with our ligand, and as you can see it is exactly same as that of the seventh fill volume. If we hide the seventh fill volume, you can see that it has also extended into the core of the protein, and it is again the potential or it is again the potential water occupying site, as you can see as that of seventh. Exactly these two regions overlap. So these are the potential water binding site uh, present inside the core of the protein, and although ligand is not occupying this site, it's okay, as the actual ligand binding site is predicted correctly by the fourth pose only, as it contains less water molecules and the ligand hydrogen donor and hydrogen acceptor moieties are greatly covered in this fill volume without gaining extra space inside the core of the protein where water molecules are actually occupying that surface, again highlighting that they are water occupying surfaces and not the potential ligand binding site. So 7th and 8th also encloses the same uh, ligand binding site, but they also include some potential internal water binding sites of the protein. So lastly, there is a ninth pose which is again on the surface of the protein and cannot be the ligand binding site. So we have got the three fill volumes that exactly predicts the binding site of our ligand, which are fill volume 8, 7 and 4. These three fill volumes uh, exactly predicts the potential ligand binding site, and these three poses have the least have moderate EPV values, and their surface volume is also uh, approximately equal, that is for 7 and 8 the surface volume and EPV values are exactly or nearly the same, whereas for the fourth one it is quite a less as compared to uh, fill volume 7 and 8. So we will consider the fourth to be our optimal ligand binding site that is predicted by our autoligand program. So the criteria for choosing the ligand binding site is that first you need to watch for the location of the fill volume. If the fill volume is located in the core of the protein occupied by the least water molecule and surrounded by the amino acids and occupies less volume and has EPV value less or in the mean lying near the mean EPV values out of the 9 poses, then you can choose that particular fill volume to be your potential ligand binding site. So this tutorial has specified how you can carry out how you can predict the ligand binding site that is uh, that is present in the protein whose ligand binding site is not actually present, or no ligand is present in that particular protein, and in that particular situation you need to predict the binding site of your protein by the autoligand program like the one I have just shown here. There is another uh, way where you can where you have your protein structure and you have the specified binding site known inside that protein structure, and in that case also you can run the autoligand program and see and provide the starting amino acid for your binding site, and this autoligand program in this situation will predict the more affinity regions within the binding site you have just provided, highlighting subsites within your binding sites, and knowledge of subsites within your binding site will help us to develop a more potential ligand that can occupy these subsites, and occupation of these internal subsites by a ligand will generate more higher binding affinity with your protein. So in the next tutorial we will go over how we can find the subsites within the binding sites of our protein that can be helpful for designing new potential and more fitting ligand within your protein. So that's it for this tutorial. If you have liked this tutorial, please subscribe to our channel and share it with your friends so that they can also gain knowledge about finding binding sites and predicting binding sites within the protein, which originally has less information about the same. So that's it for this tutorial. Thank you for watching.